Hi everyone, my name is Mohammed. I'm a PhD student of computer science at Rice University. I'm going to present Phylovar, a phylogeny-aware method for single nucleotide variation detection from large-scale single-cell sequencing data. This is a joint work between Rice University, University of Vigo, and IIT Kanpur. Given the reads from a single-cell data, we can infer the single nucleotide variations using the existing tools such as GATK, Monovar, and so on. Using the genotype matrix from this step, one can infer the phylogeny of the cells by tools such as CyFit and CyT. But we know that variant calling is not flawless. The errors from this step can propagate to the phylogeny inference, which is by itself not perfect too. To reduce errors in these two steps, we can infer both simultaneously. Here's an example to show the intuition behind phylogeny-aware variant calling. Let's say we have 4 reads from a single cell map to a genomic position. We can see all the reads contain the nucleotide A at this position, except for one of them which has T. Suppose we know the true phylogenetic tree of the cells, and we know that there is no mutation on the branches of the tree leading to this cell. Then we can say that the variant nucleotide is a result of technical errors. But if we see a mutation on the branches of the phylogeny leading to that cell, we can say that with more confidence that the variant nucleotide is a sign of a mutation. In reality, we don't have the true phylogeny of the cells. So we infer the true phylogenetic tree and the variants simultaneously. This idea has been implemented in different ways in works such as SciPhy and SCVilp. SciPhy estimates the posterior of the tree and mutations using MCMC sampling, and SCVilp infers the tree and the genotype matrix with maximum likelihood estimation. We know that both methods work well on the average size datasets where the number of cells and sites are in the order of hundreds. But when running them on whole genome single-cell sequencing data with a small number of cells and large number of sites, they face scalability issues. SciPhy takes a long running time to process the data, and SCVilp requires a lot of memory. We are interested in designing a new method that scales up to such datasets. So we wanted to simplify the scheme and get rid of the extra work that the other methods perform. For example, we know that SciPhy estimates the entire posterior distribution, and SCVilp finds the exact solution with maximum likelihood using integer linear programming. Instead of posterior distribution, we aimed at likelihood estimation, and instead of the exact solution, we decided to find the local optima using hill climbing algorithm. The likelihood function of Phylovar consists of 2 major components. The first part is the probabilistic relationship between the true genotypes and the observed genotypes affected by the single-cell sequencing technical errors. We call these genotypes library genotypes. If the true genotype is a reference allele, it is observed as a variant allele after sequencing with probability of alpha. Alpha is the false positive error rate. Similarly, if the true genotype is variant, and it is observed as the reference allele with the probability of beta, which is the false negative error rate. The second component is the relationship between the observed read counts and the library genotypes. We model this relationship using a binomial distribution where the probability of success is dependent on the library genotype. We can marginalize out the library genotype variable by multiplying the 2 probabilities: the probability of the observed read counts given the library genotype and the probability of the library genotype given the true genotype. Now we have the probability of observed read counts given the true genotype. The true genotype variable is determined by the underlying tree topology and the placement of the mutations on its branches. For example, if we have a tree like this for 4 cells, and if we place the mutation on the lowest common ancestor of cell 3 and cell 4, because we are following the infinite size assumption, both cells 3 and 4 will inherit that mutation. This gives a genotype profile for the cells at that genomic position. Then we can calculate the likelihood for all the cells at that site. We need to calculate this likelihood for all the sites to obtain the total likelihood value for the entire dataset. Here is the pipeline of Phylovar starting from the raw input data to the output. Given the read counts for all the cells and sites, first we filter out the non-informative sites. For this step, we use the statistic test that was introduced in SciPy. Next, we infer an initial genotype matrix without considering the underlying phylogeny. For this step, we can use monovar, GATK, or optimize Phylovar's model without the underlying tree. Using the initial genotype matrix, we can calculate a pairwise distance matrix between the cells, and then infer an initial tree topology using neighbor joining algorithm. Given the topology from the previous step, we need to find the best mutation placement for all the sites. Depending on the tree topology, there could be many possible placements for the mutations. In each iteration of the hill climbing algorithm, we'll calculate the likelihood here and compare it with the previous best score. To find a better topology, we change the previous topology using techniques like nearest neighbor interchange and the other tree rearrangement techniques. This iteration continues until we see no improvement in likelihood or we reach the maximum number of iterations. We notice that finding the best mutation placement was the most time-consuming part. The naive approach to find the best mutation placement for all the sites is by two for loops. One over the mutation placements and the inner loop on all the sites. This approach was really time-consuming. So we decided to improve the runtime of this step by formulating it as a matrix multiplication. We can show that the following matrix multiplication produces a matrix that contains the likelihood values for all the sites and all the mutation placements for a fixed topology. Matrix S is a matrix representation of all genotypes that all possible mutation placements can generate. For each topology, we calculate this matrix only once. Matrix O is the matrix for all the cells and sites that contains the likelihood of the read counts when the true genotype is assumed to be variant allele. Matrix Z is similar to O, but for the reference allele as the true genotype. Both matrices O and Z are calculated once at the beginning of the hill climbing algorithm. Matrix J is the matrix of all ones to negate matrix S. This technique reduced the runtime from hours to less than a second. We then simulated data using the simulator introduced in SciPy and tested SciPy, Monovar, and Phylovar to investigate their accuracy and runtimes under the following scenarios. We varied the number of mutations, the number of cells, the rate of allelic dropout, the effect of violations of infinite size assumption, and the effect of copy number variation. So we simulated 16 cells with 1,000, 10,000, and 100,000 mutations. Here, the number of sites in the simulated genome was 1,000 times the number of mutations. As you can see, Phylovar was as accurate as SciPy, but 3 orders of magnitude faster. We next changed the number of cells from 8 to 32 with 100,000 mutations in each simulated data. Here, the accuracy of Phylovar was comparable to SciPy, but it was 2 orders of magnitude faster. We also tested the effect of allelic dropout on the accuracy of the methods. As expected, as the allelic dropout increases, the accuracy of the methods drops. Again, Phylovar was as accurate as SciPy in all the cases. We observed a good correlation between the true allelic dropout and the false negative error rate that was inferred by Phylovar. Finally, since Phylovar assumes all the reads are coming from a diploid strand, we introduced extra copies to a fraction of loci to see the effect of copy number changes. As you can see, the accuracy of all methods drops with increasing the effect of copy number variations. It's interesting that when we introduce violations of infinite size assumption to simulated data, in overall, the accuracy of the methods did not change. We applied the methods to a triple-negative breast cancer dataset. This dataset contains 32 cells that are sequenced by whole exome sequencing, and after filtering out the non-informative sites, 3,375 sites were remained. This dataset was useful for accuracy measurement because the bulk data from the tumor and normal cells were available. Here are the heat maps of the genotypes we inferred using Phylovar, Sci-Fi, and Monovar. As you can see, the heat maps of Phylovar and Sci-Fi are similar, but the profiles of Monovar seem to be noisy because of not considering the underlying tree. Phylovar inferred around 18% false negative rate and around 1% false positive rate in this dataset. Next, we ran a SNPF to find the non-synonymous mutations. Among the 3,375 loci, 652 sites contained non-synonymous mutations. Then we ran GATK to call mutations in the bulk data. We found out that 84% of the mutations that Phylovar detected existed in the bulk data too. We ran Phylovar on a challenging dataset in terms of its scale. This dataset contains 16 neuron cells from a normal human brain. They are sequenced using whole genome sequencing, and Sci-Fi's statistic test identified around 2.5 million candidate loci. Here you can see the heat maps of Phylovar and Monovar on the loci with non-synonymous mutations. Phylovar processed it in around 17 hours, but Sci-Fi could not terminate the job after 10 days. Phylovar detected around 75% false positive error rate for this data, which is a very large value. We realized that among the non-synonymous mutations detected by Phylovar, 12 mutations occurred in the genes related to neurodegenerative diseases. As a conclusion, we introduced a scalable method for phylogeny-aware SNP detection from single-cell sequencing data. Phylovar is suitable for whole genome and whole exome sequencing data. As a future direction, we will investigate the possibility of deviating from infinite size assumption for these large datasets and move towards finite size models. Finally, I want to thank my supervisor, Luai Nakle, our research scientist, Hugh Ogilvy, and all my lab mates at Rice University Computer Science Department. My special thanks to David Posada and Monica from University of Vigo, Hamim Zafar and Sunkara from IIT Kanpur, and Nicholas Naven and his student Min from MD Anderson Cancer Center. Thank you so much for your time and patience to listen to this talk.